Good morning. It's my pleasure to present Dr. Terry Williams for his talk today. Terry did his MD PhD at Albert Einstein, finished his radiation oncology training at University of Michigan, as well as his postdoc fellowship there. And then he started his faculty position at Ohio State University. I've known Terry a long time during my time there as well. We've collaborated on phase 1 clinical trials. He's now Associate Professor of Radiation Oncology and the Vice Chair of Translational Research there. So, Terry.
Thank you, Dr. Wu, for that nice introduction. It's a pleasure to be here. I came here 7 years ago, so it's nice to see some of the changes that have been going on at Emory, but I'm very excited to hear about your proton center that's developing in radiation oncology. So thank you for the invitation, Dr. Reyes, Dr. Lozinski, Dr. Wu, for coming here to talk with you guys about some of the work we're doing at Ohio State. That's the slide here. So this is the outline of the talk. I'm going to kind of give some background on DNA repair and cancer therapy, talk a little bit more about radiation therapy's role in various GI malignancies, and then talk about some of the recent evidence suggesting that not every single tumor has the same radiosensitivity. There's molecular evidence for varying radiation sensitivity, which we've suspected for a long time in the clinic based on some of the clinical data we have. And then I'm going to talk about some of the work that we're doing in my lab to target oncogene-mediated radioresistance and thinking about tumor-specific approaches to guide radiation therapy for GI malignancies. So, as many of you are aware, DNA repair pathways have long been a target for cancer therapy. In the past, the therapeutic window for cytotoxic chemotherapies was largely based on the premise that cancer cells divide more rapidly than normal tissue. And so targeting the cell cycle represented a major mechanism by which our former therapies worked. And as you know, DNA damage can come in many forms, whether it's single strand breaks, double strand breaks, base damage, bulky adducts, cross-links, depending on the type of chemotherapy or other type of treatment you might use. When it comes to radiation therapy, we think that the most toxic lesion to tumor cells are the double strand breaks. So for 1 to 2 gray of radiation, you might develop 40 to 50 double strand breaks and 1,000 or so single strand breaks. But it's really the double strand breaks that are thought to drive the efficacy of our treatment. And so looking globally at DNA repair pathways, we're really dissecting out as a field what are the major mechanisms of DNA repair. Things like double strand break repair, the two main pathways being non-homologous end joining repair, happens throughout the cell cycle, or homologous recombination, which is subdivided into sort of classical HR versus alternative NHEJ and single strand annealing. And then you have other DNA repair mechanisms there, single strand break repair, base excision repair, nucleotide excision repair, and some of those others that we're starting to target. So this is a nice review that came out in Cancer Discovery. Now it's about a couple years ago, but sort of highlights the different types of DNA repair that we see in both normal cells and tumor cells. And I highlighted the, let me go back. Thanks. I highlighted in the red boxes the types of DNA repair that we think are utilized by cells to repair damage after radiation. The predominant one is probably the NHEJ repair pathway, where we see radiation or other types of radiomimetics like bleomycin causing double-strand breaks, and this being sensed by proteins such as Q70 and Q80 as part of the DNA PK complex, activating DNA PK signaling, and then eventually activating effector proteins to repair that DNA damage, like XRCC4, XLF, LIG4, Artemis. And in the HR, which is another very important pathway, as you're aware, in cancer therapy, it's used a little also for repair of radiation, maybe not as much as NHEJ or even the single-strand break repair, but includes damage centers of the MRN activating through the PIK3-like kinases like ATM, ATR, that subsequently activate RAD51 or other components of the HR repair pathway, BRCA1, BRCA2, PALB2, et cetera. So I think a lot of opportunities to target these pathways to enhance our therapies, whether radiation or chemotherapy. And reflective of that, if you look at not just at this nice review, but we're starting to see a lot of these targeted therapies that are targeted DNA repair or inhibition of DNA repair hit the clinic. So now we have a whole range of molecular targeted therapies to choose from when we're designing clinical trials. There's ATM inhibitors in clinic, ATR inhibitors, CHEK1, CHEK1/2, Wee1 kinase inhibitors, DNA PK inhibitors, basic excision repair inhibitors like TRC102. So an exciting time, I think, for people who are still actively interested in doing DNA repair or thinking about combining DNA repair inhibitors with immunotherapy to sort of enhance the efficacy of that treatment. So I'm going to switch gears, talk a little bit about what are some of the clinical questions facing radiation oncologists, particularly as it relates to gastrointestinal malignancies. And so to start off, I'm not sure, there may be many people who are not familiar with how radiation works. I'm going to talk a little bit about that and then get into the clinical questions that we're going to address. So radiation therapy, you may know, is actually the most commonly used oncologic therapeutic agent. In fact, it's estimated to be responsible for 40% of all cancer cures. And about 50 to 60% of all cancer patients at some point during their treatment course will likely receive radiation. This is data from the Institute of Medicine. And it's an effective and efficacious and cost-effective therapy. And you may know that radiation alone is used to cure many different types of patients, whether it's an early stage lung cancer or prostate cancer, early stage head and neck cancer like a laryngeal cancer, and other diseases. So in terms of gastrointestinal malignancies, of which there are obviously many, radiation plays a role in many of them. So whether it's esophageal cancer, gastric cancer, pancreatic, liver cancer, rectal or anal cancer. Today's talk, I'm going to talk focus a little bit more on pancreas and rectal cancer. So to frame this, I'll frame this sort of the challenges we're facing. I'll start talking about what the issues are in rectal and pancreatic cancer. So for rectal cancer, the ones that we give chemoradiation to prior to surgery, for example, these are patients with locally advanced disease. We're asking questions now in the clinic. Who needs surgery? Is it possible that chemoradiation is enough for patients with a careful watch and waiting approach after therapy is done? But also, what are the molecular pathways that are driving resistance to chemoradiation? Because the vast majority of our patients do not experience what was called a pathologic complete response after chemoradiation prior to surgery. And in pancreas cancer, a certain percentage of our patients, about 10 to 15%, undergo pancreatic cancer resection, but we don't know really who needs radiation or even chemoradiation after surgery, and what are those molecular pathways also driving resistance, because both rectal and pancreas cancer are thought to be clinically relatively radioresistant histologies, rectal and pancreas adenocarcinoma. So for rectal cancer, this constitutes about a third of colorectal cancer cases, and it's the number 3 most common cancer in the United States, with about 135,000 cases per year. Interestingly, the molecular biology and genetics between colon cancer and rectal cancers is actually remarkably similar. I'm going to show you some data on that. And we know that for rectal cancer, most of our therapy is typically delivered with 5.5 weeks or so of radiation, concurrently with 5-FU or Xeloda, the oral form of 5-FU. And we know that this improves local control, and improved toxicity rates compared to postoperative therapy, and improves sphincter preservation. And so the TCGA has provided us a lot of data to understand what are the molecular pathways that are important in this disease. We know about 10 to 15% of tumors are hypermutated, with about 75% of those related to microsatellite instability due to loss of MMR genes or polyaberrations. And in that 10 to 15%, as you can see here, these are the hypermutated tumors. You can see that mutation rate is much higher than the non-hypermutated tumors. And these are the typical aberrations we see in mismatch repair proteins and other DNA repair proteins. And the set of genes that are mutated between these two are fairly different. So with hypermutated tumors, lots of mutations in ACVR2, APC, TGF-beta2, and non-hypermutated tumors, APC, KRAS, P53 predominate in that group of patients. But this is what I was talking about. Largely rectal, when we talk about the non-hypermutated tumors, which again, the vast majority of tumors we see, rectal cancer shown here is virtually indistinguishable from right colon, transverse, left colon tumors on the basis of copy number alteration, DNA methylation, gene expression patterns, whereas hypermutated tumors are clearly different. The TCGA data has also defined the major molecular pathways that are aberrant in colorectal cancer, including activation of the Wnt pathway, inactivation of TGFR beta signaling, activation of PI3 kinase signaling, and receptor tyrosine Wrasse type signaling. And I'm going to highlight that a little bit because some of the work that Dr. Wu and I and others have been doing is to focus on this particular pathway, the Wrasse pathway. We see KRAS mutations in about 40% of colorectal cancer, and additional NRAS activating mutations in about 10%, and BRAF activating mutations in about 5%. So as I mentioned, we commonly treat rectal cancer with 5.5 weeks of preoperative therapy, with 5-fluorouracil capecitabine and radiation. And as I mentioned, we only see a pathologic complete response, meaning no residual tumor cells in the surgical specimen in only about 10 to 15% of cases. With surgery, we're able to attain very high pelvic control rates and 5-year survival rates. But as I mentioned, many patients are interested now in non-operative management and preserving their organ, particularly for distal tumors where an abdominal perineal resection may be required to leaving them with a permanent colostomy. So can we safely and carefully treat patients without surgery and change the standard of care for rectal cancer? And so there's a lot of publications that are coming out supporting this. So the Brazilians actually, Angelita Habergama, were some of the first to really explore this. And that actually came about as a result of lack of surgeon availability in Brazil after chemoradiation. And so patients often had to watch and wait, look for their cancer to recur until they can find a surgeon, usually months later, to take out the tumor. And Memorial published some data with a group of patients that refused surgery and showed good outcomes with this sort of non-operative management, just sort of a watchful waiting approach after chemoradiation. The Brazilians updated their experience. The Dutch and the British, this is the British Encore project, recently have looked at this as well. And a lot of our groups are starting to develop prospective clinical trials. I sit on the Rectal Anal Task Force, and we just vetted 2 large cooperative group concepts, one from Alliance, one from ECOG, that are looking at organ preservation in rectal cancer in a large hundreds of patients sort of clinical trial. And what we know is that when we give patients chemoradiation and just sort of give them the watch and waitful approach, about half the patients have actually a great response, or what's called a clinical complete response. And of these, about 30% will develop a local recurrence. So numbers are still fairly high in the sense of recurrence, but the good news is that if you can salvage them with surgery after they recur after chemoradiation alone, still the salvage rate is quite high, 90 to 95%, where you still see cure rates. And so leading to a 5-year cancer-specific survival of 91%. So a lot of interest in this, but one of the questions I'm interested in, I think Dr. Wu and others are interested in, is can we improve radiation and chemoradiation efficacy whereby we can elevate this to a point where we don't need surgery as often for patients and improve those control rates with chemoradiation or chemotherapy plus chemoradiation alone. Maybe moving some of that, what's typically given postoperative chemotherapy, moving it up front. Okay, so switching gears a little bit to give you some more background on pancreas cancer before I jump into some of our work. Pancreas cancer, as you know, really bad disease. Only about 10 to 20% of patients are able to go to surgery. And as I mentioned, radiation is controversial. I'll get into some of that data. In terms of outcomes, this is a common cancer in the sense that about 45,000 patients per year are developing this, but more importantly, it's very highly lethal. So and it's projected by the year 2030 that pancreas cancer will be the #2 cancer killer, only secondary to lung cancer. And that's in part due to rise in incidence, as well as just stagnation of our ability to improve outcomes in this disease. So what is driving these poor outcomes? So a lot of things. Inability to detect pancreatic cancer at an early stage is a big problem. Difficult surgery, high rates of metastatic progression, resistance to our current therapies like radiation or cytotoxic chemotherapy. We don't know how to select patients for therapy. Who needs radiation? Who needs heavy chemotherapy prior to surgery? a toxicity from therapy too. Can also shorten life for some of our patients. And you know, as Dr. Lazinski and others are working on, this is a relatively immune tolerant tumor microenvironment. So how can we improve the sort of anti-tumor effects of our immune system in these patients? And lastly, tumor cachexia further complicates the issue. So today I'm really going to, there's just so much to talk about in pancreas cancer, but what I'm going to focus on, some of our work is understanding resistance to radiation in pancreatic cancer and rectal cancer, and then being able to select patients for therapy. So as a background, to understand the genomics, I showed you genomics for rectal cancer. I want to show you some genomics studies from pancreas cancer. This is the largest pancreas cancer work that's been done by, actually this is the Australians, and they looked at close to 500 patients with whole exome sequencing, and they found about 32 recurrently mutated genes that sort of co-link or segregate into 10 core pathways I'm going to show you. And they also identified 4 transcriptional subtypes, 3 of which were validated from a previous study. And so right at the top, you know, we've done a lot of genomics in pancreas cancer, but this confirmed that we still know the major aberrant genes in this disease. It's KRAS, P53, INK4A, SMAD4. KRAS mutations are by far the highest, and they're all activating, essentially, mutations, mostly in codons 12 and 13, where we see 92% activation. But there's a lot of other lower frequency types of mutations in various pathways like KDM6A. I'm going to talk about ARID1A, TGF-beta-R2, MEK4, RNF43. And so some of these provide potentially other opportunities for targeting. KRAS is the core signaling pathway that's aberrant. As I mentioned, also cell cycle, CDKN2A, P53, TGF-beta signaling. These are the highest pathways, or most frequent pathways that are aberrant. But there's also fairly frequent groups of mutations in SWI/SNF chromatin remodeling type pathways. And then, of course, the DNA repair pathways and newer targets potentially with RNA processing and even Wnt signaling. Okay, so talking about the role of radiation in this disease, we've really struggled over the last couple of decades. Standard of care for resectable pancreas cancer remains surgery followed by adjuvant chemotherapy on the basis of the SPEC1 and CONCO trials, et cetera, where chemotherapy has shown a benefit for patients in improving overall survival. SPEC4 recently showed that the combination of GEM and CAPE improved survival as well, but the role for adjuvant radiation remains controversial. ASCO guidelines and NCCN guidelines still recommend consideration of adjuvant radiation for node-positive disease or positive margins, but we're still trying to figure out the role of radiation. I'll explain that. But regardless, I think if you look at the prospective randomized trials, the ones for adjuvant pancreas cancer, there's still high rates of local recurrence. You look at the older JITSIG or EORTC trials or the SPEC1 trial, the local recurrence rates were 40 to 60%. CONCO, 37%. There was a suggestion in the trial that used radiation, the RTOG trial, where the local recurrences dropped to 23 to 28%, that chemoradiation might be improving local control in a population where there was actually higher rates of positive margins. So that sort of provided justification for this current trial we have that's kind of limping along through the cooperative groups. It's more than halfway done, but it's nearly 1,000 patients on this trial where we randomize patients to, after about 5 or 6 cycles of gemcitabine after surgery, to one more cycle of gemcitabine or gemcitabine followed by chemoradiation with cytabine or 5-FU. So hopefully this will provide some prospective data to address the role of this radiation in this disease. In locally advanced pancreas cancer, where we don't take out the tumor and we just treat patients with chemoradiation, we're still stuck trying to figure out the role of radiation. And this is just as another example, this ECOG trial showed that radiation benefited patients, where patients had improved overall survival when you added radiation to GEM. But it was a small trial, only 74 patients, and it closed to poor accrual. So really an underpowered study. And then more recently, the LAP07 trial looked at the addition of radiation to gemcitabine similarly, and basically found no differences in overall survival, although there was a difference in progression-free survival that was borderline, p-value of 0.06, and they found that local failure was reduced with radiation. Still leaves us wondering, does radiation actually impact survival? So groups have been trying to say, well, this radiation wasn't good enough. We need to do dose escalation, radiation therapy. And so this study from MD Anderson, from a colleague of mine, looking at dose-escalated IMRT, showed that higher doses of radiation are better. But if you look at the local regional, local control or local recurrence regional free survival, we're still talking about 60% or more patients that have either a local recurrence, local regional recurrence, or death at 12 months. So we're still doing extremely poorly here. This phase 1, phase 2 trial from my colleague at Michigan, when he was at Michigan, who's now at Penn, added radiation to gemcitabine, full dose gemcitabine-based therapy, and basically showed pretty good outcomes compared to historical controls. But still, if you look at the 2-year freedom from local progression, we're still talking about 40% of patients still having local failure with dose-escalated IMRT and full dose gemcitabine. So it really begets the question, does radiation matter in this disease? And so this, I think, is important to address. So the classical ideology is that pancreas cancer is a highly metastatic disease. Radiation is not going to be, or local control is not going to be as important, despite the high local control rates local failure rates that I showed you. This study from Hopkins showed that about 30% of patients die of local progression in a group that was largely thought to die from metastatic disease. And also high rates of local failure, radiation dose are significant predictors of survival in pancreas cancer. So we need to identify who are the patients who are likely to have a local progression. Perhaps we should be treating those with dose-escalated radiation, selecting patients better where we can see the benefits of our treatments. And so that remains the challenge that we have. And we don't have great predictive factors currently for predicting who's going to fail distantly early, who's going to have a local predominant failure pattern. Molecular markers are controversial currently, so we need to do better. How can we improve our radiation efficacy and select patients for radiation guided approaches using molecular analysis? So that's sort of the background. Now switching gears to understand, we've long thought that in the clinic, tumors have varying radiosensitivities, such as, classically, high-grade gliomas or melanoma. These are radioresistant histologies. Also, pancreas cancer and rectal cancer, especially pancreas cancer, relatively radioresistant. But what is the molecular preclinical data that supports that? And so the whole idea of precision medicine radiation is trying to select a population that's going to benefit from therapy, right? We've done this well in tumors that have EGFR mutations, for example, or HR-deficient ovarian cancer, selecting for a PARP inhibitor. But for the radiation side, we have not selected patients very well. Dose protocols are fairly uniform. There's very little adjustment for dose and fractionation for inherent radiosensitivity. And we know that biologic heterogeneity is a hallmark of cancer. So how can we take advantage of this? There was a group at Moffitt who recently published this radiation sensitivity index called the RSI, where they generated from many, many different cell lines, close to 50 cell lines, a radiation sensitivity index of 10 different genes based on the survival of those different cell lines to 2 gray of radiation. And they validated in preclinical models and then validated in human cohorts and showed that in head and neck cancer or in rectal esophageal cancer or breast cancer, they could pick out the patients who are radiation sensitive based on this 10 gene signature. More recently, they've taken advantage of the Orion data that exists through Total Cancer Care at Moffitt, OSU, and other institutional collaboration, looking at thousands and thousands of patients with variant solid tumors and applying that radiation sensitivity index to determine what tumors are likely to be radioresistant, what tumors are likely to be radiosensitive. And just as we suspected, high-grade gliomas, for example, are very radioresistant. So to the right is radioresistant, to the left is radiosensitive. Sarcoma, surprisingly, thyroid papillary cancer, which may be related to high incidence of BRAF mutations, or melanoma, another disease where you have high BRAF mutations. But rectal and pancreas cancer are also sort of more near the top, in contrast to what we consider as relatively radiosensitive histologies in the clinic, such as cervical cancer or oropharynx, head and neck cancer, we see a lot of HPV-related disease that's quite chemoradiation sensitive, or the leukemias, which are fairly radiosensitive as well. So that seems to support a lot of the clinical, our clinical understanding of what's sensitive, what's resistant. This is another study that came out of a group who had, a colleague who was at the Broad Institute and applied a large high-throughput profiling platform for over 500 cell lines to look at radiation sensitivity. And these are the different tumor types. You can see 90 lung cell lines, 43 colorectal, pancreas was about 31 cell lines. And they grouped by histology or tumor type what was radioresistant, what was radiosensitive. And you can see pancreas is right here near the top. Large intestine, though, surprisingly, was a little contradictory, a little bit more radiosensitive in this particular study. But what I like most about this study is that they had a full gene expression data for all these different cell lines, mutational data, and they correlated molecular pathways with what was sensitive, what was resistant. And one of the top signaling pathways that was associated with radiation resistance was activation of EGFR, HER2, FGFR, AKT, RAF, MEK. A lot of the proteins that are involved in receptor tyrosine kinase Wrasse activation. So where do we stand? So, sort of the first half of the talk. Pancreas cancer dose escalation strategies have failed so far, in my opinion, and are limited by normal tissue toxicity. Solid tumors have variable sensitivity to ionizing radiation therapy, although our clinical knowledge appears to be supported somewhat in part by the preclinical data I just showed you. And we have not done a great job applying molecular profiling or genetics to select patients for radiation therapy. The radiosensitivity index is one of the first steps to be able to do that and can predict sensitivity, but we need better approaches. We also need novel molecular strategies that are going to leverage tumor dependencies and try to target and overcome tumor cell intrinsic radiation resistance. So that brings me to what I'm going to talk about, stuff that we've done in the lab to try to address some of these pathways. So as I mentioned, the Wrasse pathway, activated oncogenes, they're very common in both pancreas and rectal cancer. Basically the canonical pathway I'm sure all of you are familiar with. Activation of receptor tyrosine kinase through Wrasse activates downstream RAF kinases, MEK, ERK, translocation to the nucleus to promote proliferation survival. Also activation of parallel pathways including PI3 kinase AKT, mTOR, or RAL, protein like PLC or other types of downstream RAS signaling pathways. These are small, RAS family proteins are small GTPases, as you may be aware. They're very commonly expressed in cells. They shuttle between GDP-bound and GDP-bound inactive state and GTP-bound active state. And it's quite fast. It's been a very difficult target to inhibit through pharmaceutical approaches. There's three main isoforms, H, N, and KRAS, and these all lead to permanent constitutive activation, therapeutic resistance. RAS mutations serve as the most common oncogenes in cancer, with about 20 to 30% of all human tumors having a RAS mutation. In terms of KRAS, one of the three major isoforms, it is the most common RAS family member mutated, and as I mentioned, common in pancreas and colorectal cancer, but also in lung cancer or biliary cancer or uterine cancer. These are diseases that are commonly treated with ionizing radiation. And these KRAS mutations are associated with poor prognosis. You look at colorectal data sets, you look at lung data sets. These are examples of some colorectal and lung data sets here. The presence of a KRAS mutation induces generally worse outcomes. Also predicts for lack of response to EGFR-directed therapies, such as erlotinib, gefitinib, cetuximab, panitumumab, shown there. So I got interested many years ago in looking at the radiation response to RAS-activated mutations. This has long been studied, actually, so it wasn't particularly novel. The first study was published back in 1988, so this has been going on for a while, where a radiation oncologist, believe it or not, in Michigan, Marshall Sklar, who first transfected cells with different types of RAS plasmids, HRAS, NRAS, KRAS, and showed that RAS transformed cells are indeed radiation resistant. It was a short report, and that was basically all they showed for the most part. He showed in that paper. Since that time, the group at Penn had done a lot of work over the 90s and early 2000s to really look at targeting this pathway and inhibiting RAS through farnesyl transferase inhibitors or geranyl transferase inhibitors, and showed that you could improve radiation sensitivity by targeting this. But the molecular mechanisms of that have largely been not understood very well. We're seeing now emerging clinical data supporting KRAS mutations as being radiation resistance in lung cancer. Was one of the first groups at Dana-Farber to show this. More recently here, a group at Emory with Kristen Higgins showed that KRAS mutations associated with worse local control, patients treated with stereotactic radiation therapy. A number of reports also support in rectal cancer that presence of activated oncogenes, KRAS or BRAF, and RAS are associated with worse pathologic complete response. But how does this cytoplasmic oncogene, KRAS, affect DNA repair in the nucleus? So that's really the question that we set out to look at in my lab. So we started looking. So most of the part of this talk is going to be focused on colorectal cancer cell line. We took a couple isogenic cell models, one HCT116 or SW48 cells that have a single point mutation in KRAS, and showed in both that the presence of a single point mutation renders the cells radiation resistant. And it appears that it doesn't really matter what point mutation in KRAS is induced in radiation resistance. So using similar isogenic cells, the SW48 cells, we showed that all the different types of mutations that we commonly see are associated with radiation resistance. Now, when we took this to xenograft models, we found that indeed the KRAS mutation induces marked radiation resistance. These flank tumors were treated with a 4-gray for 5 days. We essentially cured all the KRAS wild-type tumors, colorectal tumors, but that single G13D point mutation induced marked radioresistance. So beginning to delve into this from the molecular standpoint, DNA repair, we first started looking at how do cells repair the DNA damage after radiation, looking at assays such as the mitotic catastrophe assay or neutral common assay. And we found that the presence of a KRAS mutation reduced the amount of cells undergoing mitotic catastrophe, as shown here. Here's the KRAS mutant cells. And in the neutral common assay, where you study how the DNA damage is repaired in the nucleus over the course of 24 hours per se, we find that the mutant has, interestingly, an increase in sort of that comet tail moment. But with time, there's a rapid clearance of that DNA damage. So it appears that the KRAS mutants have heightened sort of DNA repair response. And that's confirmed with a gamma H2AX nuclear foci assay, where you're looking at a nascent double-strand DNA breaks in the nuclei, treating cells with radiation, and then looking at how the wild types versus the mutants respond. The mutants are in orange, and you can see that they rapidly clear these gamma H2AX foci. And then looking at the effects on cell cycle, radiation normally induces a G2 arrest, usually somewhere between 5 to 10 hours after radiation in many tumor cells. And we can see that here. So this is the orange is 5 hours after radiation. We start to see an induction of G2 arrest. But that usually by 10 hours, cells are starting to release from the G2 arrest. And then you start to see cells cycling towards the G0, G1 phase, as shown here. In the mutants, we see similarly activation of a G2 arrest at 5 hours, but by 10 hours, we're already seeing significantly higher rates of cells released from that block and re-entering the cell cycle. Okay? And that's shown also here, tabulated here, this graph or chart showing that the mutants have higher levels of cells in the G0, G1 phase and reduced numbers of cells in that G2 arrested phase at time points like 10 hours or 20 hours. So it does appear that KRAS mutations are associated with accelerated DNA repair, but now how do we understand the molecular mechanism of that? So we performed Affymetrix RNA profiling, reverse phase protein profiling arrays to look at DNA repair proteins that are altered by KRAS. And we found 53pp1 as being this is a protein that mediates the choice between NHEJ and HR repair in cells, and found that the mutant cells had much higher levels of baseline 53pp1. This is before radiation, shown here in these two cell lines, the HCT116 and the SW48. Interestingly, we also find heightened activation of ATM and CHEK2. Moreover, when you look at 53pp1 foci in the nucleus, significantly higher rates of 53pp1 foci after radiation in the mutants, shown here in the light gray, in both of those two isogenic cell lines. So that became a target for us, and we looked at, we sort of mined expression data publicly available. We found multiple data sets showing that 53pp1 expression in cancers is higher than normal tissue. And we began to look at repair pathways. So we subdivide again, looking at double-strand breaks in radiation, which are predominantly repaired by HR and NHEJ repair pathways. We took reporter assays to look at NHEJ and HR repair using GFP plasmids. And so you can see that when you knock down KRAS, when you overexpress KRAS G13D, we actually see a reduction in HR repair in cells, whereas the opposite happens in the NHEJ repair pathway, where we see activation of the NHEJ repair pathway with introduction of KRAS G13D. And that can be attenuated by knocking it down. So if you sort of reverse complement the approach and knock down KRAS G13D, you see return of HR activity or suppression of NHEJ repair. So to us, this pointed to that this pathway is utilized in NHEJ repair as a major mechanism for repairing the damage. You can target 53pp1 if you knock it down. We find that the KRAS mutant cells are sensitized to radiation with partial knockdown using siRNA, whereas the wild type cells show essentially no difference. That's shown in both of these colorectal cancer models. And when we did the xenograft studies, we saw that, as we would predict, that stable knockdown of 53pp1 sensitized cells to radiation therapy, shown here versus here. Interestingly, knockdown of 50pp1 actually accelerated tumor growth. We're trying to understand that process, but clearly 50pp1 is mediating some of the radiation resistance in these KRAS cell lines. So we've gone to sort of begin to understand how KRAS can affect nuclear DNA repair. And so the model, a revised model, is 53pp1 is upregulated, activating NHEJ, which subsequently allows for cell survival. But what's going on in between? So how does KRAS mediate upregulation of 53pp1? So we did a promoter search of the 53pp1 promoter to look at various transcription factors associated with radiation resistance. So it's a huge list of transcription factors. So how do you begin to delve into that? Well, we were focused on NRF2 because NRF2 has been shown to be important for antioxidant response, and one of the ways that radiation works is through reactive oxygen species that you need oxygen fixation to cause damage to cells. So we focused on NRF2, and actually when we looked at publicly available expression data, NRF2 correlated well with KRAS mutations, both in colorectal cancer, pancreatic cancer, other types of cancers like uterine cancer, as you can see here. So we got interested in this. We looked at the 53pp1 promoter. There's multiple antioxidant response elements. There's 3 that where NRF2 would likely bind to. And so we performed a chip assay with the NRF2 antibody to look at 53pp, how much NRF2 is being associated with these promoter regions on 53pp1. And then we found in accordance with our hypothesis that NRF2 was, NRF2 binding to the 53pp1 promoter was much higher in the KRAS mutant cells shown in the light gray, to all 3 regions of that promoter, ARE1, 2, and 3. Likewise, just looking at KRAS mutation induces higher levels of RNA, NRF2, and protein in our model. So it does indeed appear that KRAS is inducing higher levels of NRF2, which may be mediating 50pp1 expression. Similarly, NRF2 is upregulated in colorectal cancer compared to normal tissue in similar data sets shown here. And if you inhibit NRF2 through a genetic approach using siRNA or a pharmacological approach, you can now radiosensitize these KRAS mutant colorectal cancer cells shown here with siRNA, where we also see, supporting our hypothesis that when you knock down NRF2, you see reductions in 50 DPP1 levels. And then treating cells with a pharmacological inhibitor of NRF2, brucitol, can radiosensitize KRAS mutants and cause reductions in 50 DPP1 expression shown here. Compare these two lanes here, or this to that. So it does appear that inhibition of NRF2 does indeed help to sensitize KRAS mutant tumor cells. Just another point I want to make is that NRF2 resides in the cytoplasm bound to KEAP1 for the most part. And so KEAP1 inhibits it and essentially causes degradation of NRF2 when it's keeping the pathway inactive. When you have activation of DNA damage through reactive oxygen species or electrophiles, that's when NRF2 dissociates from KEAP1, is able to go into the nucleus and cause effector downstream transcription of antioxidant genes. And we see that in accordance with our hypothesis that NRF2 is higher higher levels of NRF2 in the nucleus for the KRAS mutant tumor cells. Okay, so we're going to go back upstream and look at targeting KRAS. So if we target KRAS with siRNA, likewise, just by downregulating KRAS, we can see reductions in 50DPP1 and reductions in NRF2, sort of confirming our hypothesis that this pathway is all related in these cell lines. And that's similarly true for the mRNA expression. And then when you look at radiation survival, using clonogenic assays, knockdown of KRAS also induces radiation sensitivity. So our revised model now is that KRAS activates NRF2 to translocate into the nucleus, bind to the promoter regions of 50DP1, cause an increased 50DP1 expression, activate an NHEJ, and subsequent self-survival. So that's where we are with that particular one. We're beginning to try to understand how to block this repair pathway. And one of the things that I mentioned before is that MEK confers radiation resistance, and multiple preclinical studies have shown that targeting the MEK pathway downstream of KRAS can radiosensitize cells. So we looked at trametinib, which is a MEK inhibitor, which potently inhibits phospho-ERK, and we can see that NRF2 and 50DP1 are also downregulated in those cell lines. Where does MEK sit? It sits downstream of RAF kinases, just in front of ERK kinase. And the beauty of MEK inhibitors is that they are highly selective and have tight specificity for their targets, reducing the potential resistant pathway mutations that might develop for inhibitors that target an NATB binding site. So this sort of led us to explore this. I began to explore this years ago using a MEK inhibitor to target various KRAS mutant tumor cell lines and preclinical models. You can see this older generation MEK inhibitor, PD901, potently inhibits ERK activation and can radiosensitize KRAS and NRAS mutant tumor cell lines. Similarly, when you look at xenograft models, inhibition of MEK and radiation can effectively radiosensitize tumors cell lines, shown here, where we see increased synergistic combination of radiation and MEK inhibition in reducing tumor growth and causing increased tumor growth delay. So we became interested in what is the mechanism of radiosensitization by MEK inhibitors, and given that I only have about 15 minutes left, I'm going to kind of skip over this. We began to explore a different MEK inhibitor. It's called, as I mentioned, trametinib. It's also known as GSK212, which is approved for BRAF mutant melanoma, and showed, you can read about it, I can send you the paper if you're interested, looked at this, indeed this MEK inhibitor radiosensitizes tumor cells, but it actually works in part through abrogation of DNA repair. And that's published, as I mentioned. It appears to be related to both NHEJ and HR repair pathways. I don't really have time to get into it. but it has to do again with 53p1 as well as DNA PK. But this sort of served as preclinical data that supported us looking at targeting this pathway in the clinic. We had multiple trials come out from a lot of the preclinical data. A separate group at MD Anderson is exploring the combination of trametinib and chemoradiation for KRAS mutant non-small cell lung cancer. At Ohio State, we used that preclinical data to justify 2 trials, one for brain mets and radiation, which unfortunately recently closed due to poor accrual. And then the trial that I'm going to talk about is the trial that Dr. Wu and I and a colleague of ours, Dr. Wuthrick, started when we were at Michigan. That was to use a MEK inhibitor in combination with neoadjuvant 5-FU radiation for KRAS, BRAF, and NRAS mutant rectal cancers. There was additional preclinical data to support our concept. This came out from Deb Citron's lab at the NCI, where she combined MEK inhibition with radiation and MEK inhibition with radiation 5-FU and showed maximal tumor growth delay in those models. Our trial schema is as follows. This is a phase 1 trial where we gave patients trametinib, the MEK inhibitor, for a week. We had biopsy tissue before, and we did a research biopsy at the end of the week to look at downstream effects and start looking at pharmacodynamic effects of inhibition. We treated them with the MEK inhibitor, 5-FU radiation. They went on a break for 6 to 10 weeks before they went to surgery, and then may or may not have had additional chemotherapy after surgery. So these are the dose levels. We started at dose level 1, a low dose of 0.5 milligrams, went on to level 2, jumped over 2A because we didn't need to do that dose level, and finished at dose level 3, where the maximum dose of trametinib, which is used in BRAF mutant melanoma, of 2 milligrams. And I'm not going to go in great detail about the toxicities, but for the most part, this was a very well-tolerated therapy. The addition of a MEK inhibitor did not seem to cause substantially increased toxicity on the standard of care backbone. We did see things like more lymphopenia, generalized skin rash. There was one patient who had to discontinue trametinib after 2 weeks of therapy as a result of the skin rash. And then for 2 patients, it was held for the last 3 days of therapy, which is really not very much. There was one DLT, diarrhea, but that was attributed to chemoradiation. And we saw the treatment response that was promising. I'm not going to say it's a home run, but promising for us. Looking at the sort of the maximally tolerated dose cohort, or that 2 milligram cohort, where we think we want to move forward into the next phase of our trial, we had 12 patients in that group, and there was a suggestion that there was a higher rate of pathologic complete response and high rates of tumor downstaging of 75%. An additional 17% of patients had a near path CR. So if you remember, traditional path CR rates in rectal cancer are about 10 to 15%. So this is encouraging. We want to bring this forward into a phase 2 trial. This is some of the pharmacodynamic biomarker analysis that we did in my lab from samples from this trial. You can see the high levels of phospho-ERK in the pre-treatment tumors by the brown staining. We used quantitative immunohistochemistry using a vector microscope with Inform software. And after 1 week of trametinib, we saw pretty substantial downregulation of phospho-ERK levels, or indicative of suppression of ERK activation. And that was correlated very nicely with the dose of trametinib that we were given. So as we increased dose of trametinib, we saw a better turning off of ERK activation. And so where we are with this now is trying to, we were actually, Dr. Wu and I are going to be presenting next week to the NCI GI steering committee to add this arm of the MEK inhibitor arm to a large NRG trial, GI-002, that's testing novel radiosensitizers in locally advanced rectal cancer. Currently, the standard arm is full FOX for 8 cycles, followed by radiation and capecitabine, then they go to surgery. With the first experimental arm that's ongoing is testing the addition of a PARP inhibitor to chemoradiation. There's actually another arm here where immunotherapy is being tested. Now that's it's Pembro, right? It's Pembro, PD-1 inhibitor, and then so this is our proposed arm that we're going to be adding to the trial. And because we did phase 1 trial with 5-FU, we're going to try to maintain that. So the conclusions so far is that KRAS mutations induce radioresistance in part through this KRAS NRF2 53BP1 pathway, and that target and downstream with MEK inhibitors can effectively radiosensitize cells in our preclinical models, and we think that's due to inhibition of NHEJ HR repair activity. I had to really gloss over that data in the interest of time. And so far in our phase 1 trial, we think that targeted MEK1/2, which inhibits NRF2, 50DP1, and other repair pathways potentially, appears to be well tolerated and has early signs of clinical efficacy. So we're excited about that. We'll see what happens, and hopefully Dr. Wu will update you in the future about where we are with that. So in the last few minutes, I want to talk a little bit about our molecular profile in pancreas cancer. So I mentioned that there's high rates of local failure. We don't know the role of radiation in pancreas cancer. How do we pick patients out who might benefit the most from radiation? So we chose to take an approach by looking at microRNAs, which, as you know, are intracellular RNA molecules that function in post-transcriptional gene regulation. They function by binding to cognate messenger RNA molecules and causing degradation of the message. And they can be mirrors can be oncogenic or tumor suppressive. And so what we did is at Ohio State, we took close to 100 patients who had Whipple resection, isolated their tumors, isolated the RNA out of them, and performed nanostring profiling. What nanostring profiling is, for those of you who may not be aware, is a way to look at the whole microRNAome, about 800 microRNAs. And it's a very effective omics strategy for formalin-fixed paraffin-embedded tissue. It has high specificity, sensitivity. It's automated. There's really no need for technical replicates. You don't need a lot of RNA for this. And so we profiled all 800 mirrors in about 100 patients. So we ended up with about 93 patients. And these are patients that all received surgery followed by gemcitabine-based, mostly gemcitabine. There was about 14 patients who had gemabraxane, adjuvant chemotherapy, no radiation. And you can see here, we carefully, with the radiologist, looked at patterns of failure and identified which patients had local regional recurrence, which ones had distant or both. Interestingly, almost about a quarter of patients had a local regional predominant recurrence pathway, meaning that they didn't met out until much later, and so their biggest failure pattern was local regional predominant. And so we used ElasticNet to look to sort of develop a molecular signature. I don't really have time to explain the statistical method very well, but it basically assigns a correlative weights for each of the microRNAs and associates it with them local regional control. And then we also started with the OSU cohort. We did some validation on the TCGA cohort, and we came out with. By the time we started with 800 mirrors, only after you do all the filtering and normalization, we only ended up with about 200 mirrors that were different or altered in pancreas cancer. So out of those 200 mirrors or so, we came out with 4 microRNAs that were significantly associated with local regional control. And when you combine them into a weighted correlative, sort of a complicated statistical formula, we found that in our training set that there was high risk of local failure with a certain signature of these 4 microRNAs. So this may begin to address, how do we predict which patients are going to fail local regionally, and which patients may therefore benefit most from radiation. What was most surprising for us is that those patients who had high local regional failure also had a high risk, had worse overall survival. So it seems that if you can pick out which patients have a local regional failure predominant pattern, it actually impacts survival. And so we did multivariate analysis and showed that the risk score for the 4-mirror signature indeed was still significant when you incorporate other standard clinical variables for pancreas cancer, T-stage, N-stage, postoperative CA19-9, tumor grade, margin status. It wasn't significant in overall survival when you did multivariate correction, but when we went to the TCGA data, we found, again, that the signature predicted for worse overall survival, shown here. These are all patients or patients without radiation, so which are the majority of patients. And on multivariate analysis of the TCGA cohort, it likewise was significant, remained an independent predictor of overall survival. We have since validated this. We got another data set from Seoul National University in Korea. They had a large patient data set, similar of 100 patients with all microRNA profiling. Unfortunately, they don't have patterns of failure, so we're asking them to go back and look at what is their local regional recurrences, distant recurrences, et cetera. But we were delighted to see that in this additional data set, you had a high risk for mere signature that predicted for local regional failure associated with worse overall survival. So our next steps, we're working with colleagues at MD Anderson and Mayo. They've recently sent us some samples to start to isolate RNA. Thankfully, MD Anderson and Mayo have full annotated patterns of failure for these pancreas cancer patients. So what we're going to do is try to validate that this signature predicts for local regional failure and overall survival in those additional data sets, because that's critical, to be able to identify that. So we're excited about this work. That's where I'm going to end for today, and hopefully, we'll make some progress over the next few years on these projects as well. But wanted to acknowledge the people who did the work. It's so critical. My funding sources on the left. Lin Yang was the postdoc who did a lot of the KRAS NERF 53BB1 data. Ryan did a lot of the phase 1 clinical trials work on the OSU 12054 clinical trial with the MEK inhibitor. Adriana Estrada, a postdoc in the lab, did a lot of the mechanistic work to assess how MEK inhibition is affecting DNA repair through NHEJ or HR repair pathways. And then Patrick, Amy, and Steve were involved in the microRNA project. Dr. Wu remains a fantastic collaborator, both here but before, along with my other colleague, Evan Wuthrick. And then some of the surgeons and pathology team who helped us out. So thank you for the opportunity to present to you today. I can take some questions.
Really nice talk, and I really like the molecular profiling of things that are downstream of Wrasse and your ability to modulate those pathways with siRNA knockdowns and such. What struck me is the radiosensitivity in that setting for single dose isn't really that impressive. It shifts by about 1.5 gray after a single fraction. But your in vivo mouse model data is pretty good. I mean, you separate the survival curves a lot. So do you think it's the fractionation of radiation that's important, multiple doses given over time? And given that you have a good understanding of the kinetics of DNA repair pathway with Wrasse activating mutations, is single daily fractioning the best way to give radiation, or should we be giving it every 10 hours? I know Wally would like to have his team get up at 2 a.m. in the morning to give initial fraction.
Yeah, great question. So I think that the majority of the radiation resistance induced by KRAS, and we have some data, and I didn't show you at all, for BRAF mutations, appears to be more predominant at a higher dose per fraction once you get beyond 1.8 to 2 gray per day in our cell lines and models. So that xenograph data I showed you was with 4 gray times 5, and so we found significant splitting of the curves with higher dose per fraction. And if you look at the data from both here and lung SPRT and Dana-Farber, I think that's where we're going to see a lot of where KRAS mutations are more associated with radiation resistance. We see it in rectal data sets too, where we give 2 gray per day, but the data is not as strong as it is in other data sets where you're given higher dose per fraction.
What about the timing interval?
The timing interval we don't have a lot of data on to look at multiple doses per day, but I think that if I were to speculate, I think actually it would be worse for KRAS tumors, because I think that these cell lines can repair their DNA pretty quickly, and I just don't think there would be as much a difference if you were to dose twice a day versus one larger dose per day. So. Greg.
Great talk, and nice to see the progression of your data. I had a question, since we're sort of caught in this mess as to cancer-associated fibroblasts and stellate cells, particularly in pancreatic cancer. Are they good? Are they bad? is there any understanding of whether or not they're contributing to radioresistance in this disease, or are you moving in that direction in the future?
Right. So I think a lot of the tumor microenvironment is very critical in pancreas cancer, as you're well aware. Limiting access to chemotherapy, promoting survival, making an immune tolerant state. Certainly that's something we're very interested in looking at, the role of stellate cells, fibroblasts in supporting a chemoradiation resistant tumor milieu. But I think there's some data on that preclinically, which I can share, but not a lot yet. So.
Do you think they're killed by the exposure to radiation, or do you think they sort of...
You know, they are fairly resistant. The mesenchymal cells, you know, the tumor microenvironment, you know, sort of the fibroblast compartment, stellate cells. But what they may be supporting, you know, what they may be supporting is secretion of cytokines and or other types of factors that promote survival in the adjacent tumor cells. That by using radiation, perhaps maybe you can turn some of those signals off, sort of the paracrine signaling. Okay, thank you very much.